Good morning, everyone. I am K. Harshit and I am going to talk on the topic theuropatic cloning. Theuropatic cloning is the transfer of nucleus materials isolated from a somatic cell into an enucleated osteocyte in the goal of deriving embryonic cell lines with the same genome as the nuclear donor. Therapeutic cloning refers to the use of SCNT to reprogram somatic cells into undifferentiated cells for different therapeutic purposes such as the treatment of degenerative diseases or to correct genetically predisposed conditions. The major advantage of using SCNT to derive the production of undifferentiated cells for the therapeutic use is that autologous cells can be generated. These cells would be fully compatible with the patient and would not need immunosuppressive treatment. If used in graft therapies, exciting results with therapeutic cloning has been obtained in the mouse model. For instance, SCNT embryos from the mice carrying a genetic immunodeficiency were used to establish embryonic stem cell cultures. These cells were genetically modified in vitro and then transferred back to the same animal to correct the immunodeficiency. How therapeutic cloning works. Therapeutic cloning is something referred as to somatic cell nuclear transfer or simply SCNT. SCNT involves removing the nucleus of an egg cell, replacing it with the material from the nucleus of a somatic cell, and stimulating this cell to begin dividing. This egg cell is never fertilized by sperm, and the genetic material within the cell is virtually identical to the genetic material extracted from the skin or the other cell. Once the cells begin dividing, stem cells can be extracted from it 5 to 6 days later, just as they can be extracted from embryos created through in vitro fertilization. These stem cells or the specialized cells derived from them will be a genetic match to the patient. Therefore, the promise of SCNT is that the patient's body would accept these cells after transplantation. What is the purpose of therapeutic cloning? Therapeutic cloning is to generate and direct the differentiation of the patient-specific cells lines isolated from an embryo not intended for transfer into utero. Therapeutic cloning through the production of these autologous nuclear transfer embryonic stem cells offers great promises for regenerative and reproductive medicine and in the gene therapy. As a victory of these gene delivery, this review focuses on the recent breakthroughs in the research based on therapeutic cloning, their feasibility, and their potential applications in medicine. The second part of this review discusses current roadblocks of therapeutic cloning, both in science and biomedical ethics, as well as the main alternative for therapeutic cloning. Thank you.